So you're in the Queen's Medical Research Institute, which is one of the research buildings of the University of Edinburgh. We have 3 research centres based here. On the ground floor, we have the Centre for Reproductive Health. We have the Centre for Inflammation Research in the middle. And then we have the Centre for Cardiovascular Research on the top floor. But on the site, we've got other research institutes and centres as well. We've got the Scottish Centre for Regenerative Medicine across the other side of the hospital. And then we've got the Centre for Clinical Brain Sciences and part of Edinburgh Neuroscience, the Centre for Neuroregeneration, based in the Chancellor's Building. So we have 2 speakers for you tonight. We have Professor Malcolm Macleod, who's a clinical clinician. So he sees patients with stroke. Malcolm's based in the Centre for Clinical Brain Sciences and also associated with the Centre for Cardiovascular Science. And he's going to tell you about what happens in stroke and what it is. And then we have Professor Karen Horsburgh, who's from the Centre for Neuroregeneration, and she's going to tell you a little bit about the type of research that we can do into brain conditions such as stroke, for example. So thank you very much. Malcolm is saving brains, developing effective treatments for stroke.
Well, thank you, Karen, and thanks very much, everyone, for coming out on what's becoming a colder December night. What we want to do this evening is I'll give you a little bit about what stroke's like from a doctor's point of view, a medical doctor who treats patients with strokes and tries to be involved in testing new treatments. And then Karen's going to talk a little bit about the work that goes before that in the laboratory and how we develop those new treatments. So in the first half, a clinical neurologist, and in the second half, a laboratory scientist. But I thought I would start by just going in a little bit into what a stroke actually was, because some of you might not be so familiar with this. It's an interruption to the blood supply to part of the brain. And then that stops that bit of the brain from working properly. And the consequences of that for the person having the stroke depends on the part of the brain that's affected, because the function of that part of the brain is limited. And in a warning stroke, something that we call a transient ischemic attack, or a TIA, that's a temporary process and the recovery comes back. And if the blockage is permanent, that part of the brain dies and that function can't come back to that part of the brain. But when this happens, other bits of the brain can sometimes learn to take over some of the functions which have been lost, something which happens very well in young patients and less well as patients get more elderly. So how can we think about this? There's a couple of important things which are relevant to what we're going to talk about. The first is the way the brain's put together, and Karen, who's talking next, was saying that her daughter's going to art school. I never went to art school, so this is my representation of what your heart looks like. And this little railroad of blood vessels in the middle of the brain. So what happens is that there are actually 4 blood vessels, 2 at the front and 2 at the back, which supply the brain. And then from there, blood vessels go off to supply the thinking bits of the brain around about the edge. And what that means is that if you block one of these blood vessels in the neck, then the 3 other blood vessels can take up the burden of that, and so that doesn't cause a stroke. It just means the blood finds it a bit more difficult to get through. It's like come Independence, if you were to block the M6 at Carlisle, you could still get up through Berwick and still get to Scotland or out of Scotland, depending on what you thought about independence. But if you get a problem out in one of these arteries out here, then that part of the brain can be affected permanently. So if you've got a blood clot or something in your heart that then spreads from the heart, it might move up through those blood vessels and then lodge in part of the brain up here, causing the bit of brain supplied by that artery to die. So let me just show you that again, because I made my dots too small. The dot there moves up through the blood vessels, gets through the neck fine, lodges up in the artery in the neck and causes that bit of the brain to die. And so what we see here is a scan where the blood vessels in the head have been shown up, in white, and you can see that in this area here, there's a blood vessel which is missing because it's been blocked, because there's a blockage just where that blood vessel starts off. And then the effect of that, a couple of days later, shown in this other scan, is that dark area of brain which has died as a result of the stroke. The second thing that you need to know about the brain and about what happens in strokes is that there are more energy stores in the brain, really. And so brain cells working require a continual supply of energy. Here's my favourite supply of energy, but a continual supply of energy coming down the conveyor belt of the blood vessel to keep working. So once you stop that conveyor belt and block the blood vessel, then there are very little energy stores there that can keep the brain going for any length of time at all. Stroke's a huge problem. About 5.5 million people a year die around the world from stroke. That's 1 every 6 seconds. It's about 30 since I've started talking. 1,500 strokes per year, 150,000 strokes per year in the UK, and roughly 40% of patients will die within the first month or so as a result of their stroke, and 30% will become disabled, will need other people to help them with washing and dressing and cleaning as a result of the stroke that they've had. Who gets stroke? Well, here are some risk factors for stroke: age, high blood pressure, diabetes, irregularity of the rhythm of the heart or problems with heart valves, furring up of the arteries in the neck where the fur can break off and go up to the brain, furring up of the main artery from the heart, and whether it runs in the family, and whether or not you smoke cigarettes. And in younger patients, there are other less common causes: vessel splitting, or having sticky blood, or having inflammation in the blood vessels. And we used to think that having holes in the heart was important for causing strokes, but now that's less clear. And how do we treat it? Well, I went early on through the things that cause a stroke, and these are things that potentially we can treat. So if there's an interruption to the blood supply to part of the brain, by unblocking the blood supply, can we rescue that bit of the brain? Clot-busting treatments. If the blockage is permanent, that part of the brain dies, but are there things that we can give, drugs that we can give, that slow down or stop that process of brain death, perhaps until we can open up the artery again to allow that part of the brain to survive? And finally, I've said before that when a stroke happens, other bits of the brain can sometimes learn to take over some of the functions which have been lost. So there's a question about whether we can promote that process, whether we can encourage the process by which other bits of the brain take over. And I'll go through some of these in turn. But first of all, I think people think of stroke as being a terrible, terrible thing, but there's lots of good news around about stroke. And this is part of the good news. This is death rates in Scotland for stroke, for men and women. I think men are blue and women are red. And this is the death rates from 1950 to 2010, and you can see that they fall to about 1/10 of what they were before. So huge reduction in death rates in patients under the age of 69, but even when you look in the older age group, these are people 70 to 79, the rates have fallen again by about 90%. And this isn't because people are having strokes and they're surviving them better. It's mostly because there are far fewer strokes happening in the first place. So why might that be? Well, because we've got really pretty good at dealing with some things. We've got very good at dealing with blood pressure, with diabetes, with irregularities of the heart. This valvular heart disease was caused by something called rheumatic fever, which used to be really, really common, and now you hardly ever see at all, and that's one reason why strokes have dropped a lot. And cigarette smoking has fallen substantially in Scotland. It could fall a lot further. If you were to have a stroke, the one thing that you could do to reduce your chances of having another stroke would be stopping smoking. And that one thing on its own would do more than all of the other things that we in medicine can do combined to reduce your risk of stroke. That's how important it is. So we've got some treatments. We've got some treatments which work. First of all, treatments that get rid of the clots in the wrong place. So this chap is Peter Sanderco, who's one of my colleagues here in Edinburgh. And he did a large trial called IST3, where they took 3,000 patients who'd had a stroke, and they gave half of them clot-busting treatment and half of them best standard care, and showed that there was a substantial improvement in outcome in people getting the clot-busting treatment. One of the things that can happen when you have a stroke if you're immobile is you can get clots in the legs, in the blood vessels in the legs, which can break off and lodge in the lungs and cause problems, called a pulmonary embolism. And this chap, Martin Dennis, also one of my colleagues in Edinburgh, did a study called Clots, where he looked at these things, which are called intermittent pneumatic compression stockings. And what they do is they blow up and down 2 or 3 times a minute, like a blood pressure cuff, squeezing the blood out of the leg and then letting it fill up again. And what he showed in a study that just reported this year was that by doing that, you could prevent 1 death for every 50 patients who have a stroke. So a huge beneficial effect by dealing with clots which might be in the wrong place. Then there's some other treatments here, so good stroke care and rehabilitation in stroke units. Peter Langhorne, who's at the University of Glasgow, showed a substantial benefit if patients were looked after in the right place because their medical care was better. Julie Bernhardt is a physiotherapist in Melbourne in Australia, and she's running a trial called AVERT, where we try and rehabilitate patients on the first day after the stroke, rather than leaving things to settle for a day or two, which is what used to happen. And she needs to get to 2,000 patients. She's got 1,700 last time I looked, including 24 that we've got her from my hospital in Forth Valley. So within a year or two, we'll know whether very early rehabilitation works. And this chap is Keith Muir, who's a professor at the University of Glasgow, and he's doing a clinical study of stem cells in stroke, very early stages, only 11 patients treated, but the results so far seem to be quite promising. I think Karen's going to talk a little bit about that later. And then the other thing that I talked about was whether we could do anything to save brain tissue. As I said, I'm interested in testing treatments in humans that might work for stroke, and so we looked to see whether there were drugs about that might improve outcome, that we could test in a clinical trial. And we found over 1,000 drugs that had been developed in experiments in the lab, either experiments on tissue slices or cell cultures or in whole animals. And of these, 600 had been tested in whole animal models of stroke. Almost 400 improved the outcome in those animal models. So you would think there'd be a rich variety of things for me to test. In fact, 97 of them had been tested in human clinical trial. 18 interventions had been tested in human clinical trial without ever being near an animal experiment. And what we found was that only 1 drug was effective that had been tested through this laboratory paradigm, these laboratory models, and that was the clot-busting treatment I talked about earlier on. And in fact, that was used in human stroke because it worked in human heart attack, not because of any effect it had in the animals. And these 3 treatments here are stroke unit care, the humble aspirin, and in very selected patients, operations to remove the pressure inside the head with an operation called a decompressive hemicraniectomy. So what's going wrong? This process of taking drugs that work in animal models and using them for human health is called translational medicine. And here's a road sign from Wales, and you'll see it says no entry for heavy goods vehicles, residential site only, and this is the translation, except it's not, because what this says is, I'm out of the office, if you need anything translated, can you send it to someone else? And so there's a problem with the way we translate things in medicine, because we don't always understand completely what we're doing. Here's a great example. You might have seen things in the press about being able to do scans on people's heads and find out what they're thinking, or find out which bit of the brain looks after which bit of function. They're called functional MRI scans. And these people who were based in Boston had a scanner, and they did these kind of experiments, and they started to get a bit skeptical because they weren't really sure of what they were seeing. So what they did was they did an experiment with a salmon. And they put a salmon in their scanner that weighed about 4 pounds, was 18 inches long, and was dead. And then they gave the salmon one of these thinking tasks to see if they could get a signal out, see if they could. So the salmon was shown a complex, open-ended mentalizing test. They asked it to do something. They showed it pictures of human beings who were happy or sad or angry or distressed, and they asked the salmon to decide which emotion that person was experiencing at the time. And lo and behold, they found little bits of the brain which lit up, suggesting that these bits of the brain became more active when the salmon was trying to complete this task, even though the salmon was dead. So what they said was either we've stumbled on a rather amazing discovery in terms of post-mortem ichthyological cognition, dead salmon thinking, or there's something a bit off with their statistical approach. And they got an Ig Nobel Prize for this in 2012, and quite rightly so, because many people had blindly used this technique and interpreted these findings to mean something important when they meant not very much. So is there a problem with the research that we're doing in stroke that might be similar? Well, this is really how we do stroke studies. We take a group of animals, a group usually rats, but other rodents, mice, rabbits sometimes, and we block one of the blood vessels supplying the head. So we try and do to the animal exactly what happens in a human stroke. And then we divide the animals into two groups, one group of animals which doesn't get any treatment at all, and one group of animals which gets the drug that you're interested in finding out whether it works, whether it improves outcome. And then we'll wait a while and we'll measure the outcome, and that outcome might be a mouse behaviour, and we'll see more of that later, or it might be looking, for instance, at a scan to see the size of the stroke in the animal. And there are a whole lot of things that could go wrong with this, because when you divided these animals up, if you did it non-randomly, there might be something different about these animals before you even gave them a treatment that would influence whether or not the drug seemed to work. If you really wanted the drug to look good, you might select the healthy, perking-looking animals for your treatment group because they'd already be going to do well. And when you came to measure the outcome, if you knew the group that the animal belonged to and you wanted the drug to do well, then you might exaggerate, be kind about the animal's performance, so you would overestimate the outcome in that group of animals. And so we've done a bit of looking at this, and these are 7 different risk of bias items, like not doing that randomization, not doing that blinding. And this is a drug called FK506 that I did part of my PhD on more years ago than I care to mention. And this is how good it is. And in studies at high risk of bias, not randomized, not blinded, it improves outcome hugely. And in studies at low risk of bias, it doesn't improve outcome at all. So it's really important in this research, as with every other kind of research, that the people who are doing it do it properly. But animal studies and human studies can agree if they're done properly. This is a timeline with nothing on it yet. There's a drug called Neuroregeneration, which was tested in stroke studies in animals and in humans. In animals, it was given on average 10 minutes after the stroke started. In humans, it was given on average 300 minutes after the stroke started. And it didn't work in humans. That's perhaps not surprising. The clot-busting treatment TPA in animals given about 90 minutes after the stroke started, in humans given an average of 90 minutes after the stroke started. And lo and behold, when you look at how that effectiveness falls away over time, the first 90 minutes, the second 90 minutes, the third 90 minutes, in animal studies and in clinical studies, you could just about put those two lines one on top of the other. So I think if you do animal studies properly, they can be a really useful guide to the sort of treatments that might work. I just want to end by telling you a little bit about the study that we're involved with of brain cooling for stroke. Cooling the brain seems to work in patients who've had brain damage because the heart stopped beating, who've had a cardiac arrest. And there's lots of stories of people who've fallen through the ice or drowned or been stuck under a glacier or something, who should really be dead, who aren't. And we think they're not dead because their brain was cooled during the time of the injury. Cooling is so good in animal stroke studies that in many laboratories they use it as what we call a positive control. If the investigator, if the scientist can't get hypothermia, brain cooling to work, then there's something wrong with what they're doing, because brain cooling always works in animals. And finally, early results from small studies in stroke are very encouraging. So we're doing a study called EuroHype 1, funded by Chest, Heart and Stroke Scotland and the FP7 European 7th Framework Programme. And essentially what we do is put these cooling pads onto the body, and then cold water is passed through these, and as it passes through, it draws heat away from the body, and by drawing heat away from the body, draws heat away from the brain. We're looking for 1,500 patients. We've got 60 recruiting centres across 20 countries, and so far we've included 4 patients. So we are 0.3% of the way towards our recruitment target, and more effort is required. But last week we got ethical approval in the UK for running the trial in Scotland and in England. And there are 2 Scottish recruiting centres, and this is the team of likely lads, a German, a Dutchman, a Swede, a Dane and a Scot who are running this. So before I move on to Karen, I'd just like to thank all of these giants on whose shoulders I stand, and the people who funded the work, including a special mention for Chest, Heart and Stroke. Actually, not only have they funded our work on cooling, but many of the other things like stroke unit care and thrombolysis, they were instrumental in providing the funding to set up those studies in the first place. And so now over to Karen, and thank you very much.
Thank you, Malcolm. So, as Malcolm says, I'm a laboratory scientist, so I don't work with patients. I work with very small creatures, which I'll tell you about in a minute. But what I want to tell you about is how we've used animal models to really provide a better understanding of how stroke damages the brain. And by making progress in this area, how we can try and develop treatment strategies to work towards preventing the damage that occurs after a stroke. So I'm going to look at how we can protect the brain against the effects of stroke, but second to that, also look at ways that once that damage has occurred after a stroke, how we can look at ways that we can promote healing of the brain, look at repair and recovery of that brain after the stroke has happened. So, unlike Malcolm, as I've said, who sees stroke patients, I work mainly with animal models of stroke. And one of the most common models that are used are rats and mice. And although these are a lot smaller than human, their brains are a lot smaller, they actually share similar features to human brain in that their blood vessels are very similar in structure. They have similar circulation, similar blood supply, and so that lends them to studying stroke in rats and mice. And so in humans, as Malcolm has explained in stroke patients, the stroke normally occurs through blockage of a vessel leading into the brain or actually within the brain itself. And so this reduces blood supply to a particular brain region and damage ensues. So we can mimic this type of stroke in animal models by basically blocking off a vessel that lies within the brain, and this reduces blood supply to those particular brain regions. And so the stroke can be thought of as a brain attack, very similar to a heart attack, where we block a blood vessel going to the heart, reduction of oxygen and glucose, there's a heart attack. In stroke, we're blocking off blood supply to the brain. That leads to brain damage. And that blockage of the vessel very quickly leads to a reduction in blood supply. And this, I wanted to highlight, this is a type of imaging that we use to look at blood flow reductions. On the right here, this is where a vessel has been blocked and there's a marked reduction in blood flow. And in the left here, this is normal blood supply. And I wanted to show you, we've got a cool way of looking at blood flow in the brain in our animal models. And so this is a type of imaging called laser speckle imaging. I'm going to stop it there. This is real time. We can look at blood flow in the brain. And this is looking down on the top of a mouse brain. And this is a big vessel running down the middle, and it feeds off and it supplies lots of little vessels running off. And so we can measure blood flow in real time using this system. And what I wanted to show you is we're going to stop the blood flowing to the brain in a minute, and you'll see that very quickly the blood flow goes down, and that's exactly what happens when you have a stroke. The blood flow is cut off, and then if we remove that blockage, you should see a restoration of blood flow with time. So there the blockage is taken away, we see blood flow going back to normal levels. So this is the type of approaches that we might use in our experimental models to look at blood flow. This is one of the new approaches that we use. And we can also use very similar imaging techniques that's used to look at brain damage in stroke patients. So we have scanners similar to those used to look at brain damage in stroke patients, where we can look at changes in the brain in our rodent models. And so these are slices through a mouse brain, and the white bit here is where there's been some damage to the brain. So we can look at these changes with time. And then at post-mortem, we can slice the brain and look at areas of tissue damage. So we can use lots of different stains, but this is one stain that ideally shows healthy brain, living brain versus damaged brain. So the normal healthy brain is stained red here, where there's a bit of tissue damage, it's stained white. So these are the types of approaches that we've used to show that blood flow goes down very quickly after a stroke, and then after that, ensues brain damage. Now, these types of models were very important in showing that after a stroke occurs, the damage does happen very quickly in the brain and within a matter of minutes. So these are cartoons of a mouse brain here, and the black is the tissue damage. And this is to illustrate that within a few minutes there's a little bit of tissue damage, and that grows very quickly over a few hours, and within a day that tissue damage is maximal. So that means that in terms of trying to protect the brain against the effects of stroke, we need to try and get drugs in pretty quickly. And these types of studies are very difficult to do in stroke patients, of course, to really see the temporal progression, the way that the brain damage evolves over time. We can only really map that out in experimental models. And so in stroke, normally what would happen is there would be blockage of a vessel in one side of the brain, and that's often associated with weakness in one side of the body, often associated with weakness of maybe a leg or an arm. And that's because the part of the brain that's damaged is involved in motor abilities or movement and coordination. Well, when we induce experimental stroke, often in the animals, this is associated with weakness, maybe of a forelimb. And so we can study this in our models too. So if you bear with me a minute too, I'm going to show you I was going to show you a simple task, if I can get it loaded up. This is called a mouse cylinder test. And what we're looking for here is we're going to place the mouse into a cylinder, and there's a video camera recording the movement of the mouse. You can see it sniffing around, having a peek about, trying to find out what's going on, and it should stand up hopefully in a minute if it's inquisitive at all. Yep, there it goes. Stands up, pushes against the glass to see what's going on. And what we're looking to see is, is there maybe a preference of one limb versus the other? Is there any sign of weakness in this particular animal after a stroke? But the point I'm trying to make here is that in these models we can look at functional changes in relation to the types of brain changes that are going on in the stroke models, so we can make associations between them. And there are obviously lots of other tests that we can use. That's quite a simple test because it only takes a few minutes and it's not very stressful to the animals. So we use the experimental models to map out changes in the brain, and what we're trying to uncover are the types of harmful changes that occur in the brain after that stroke has occurred, because by identifying them we might be able to find treatments that can target these harmful molecules and reduce the extent of stroke after it's occurred. And the way that we'd maybe put a helmet on if we go out riding a bike, I don't know if anyone's still into riding bikes, certainly I'm past that point, but if you put a helmet on, you fall off, you're basically trying to protect your brain against the effects of that injury. So that's what we're trying to do in stroke, is to prevent that brain damage occurring and growing. So to really understand how a stroke might affect the brain, we need to think about how the brain works normally. And so each of you here, whether you're sleeping or listening to me, you all require some fuel, some energy to keep your brain cells ticking over. So as Malcolm says, we need a supply of energy, not always in the form of cookies. It's usually oxygen and glucose that's delivered to our brain, and it's delivered through a network of blood vessels. And so this is a cartoon to highlight the main brain cells, or neurons as they're called. And these need lots of energy to be active. And that energy is delivered through the blood vessels. And these cells are communicating to one another. They talk to one another in a very highly intricate way by releasing chemical messengers which transmit information from one cell to another. Now, the main chemical messenger in the brain is called glutamate. And the reason I'm labouring on that is that one of the very first discoveries that was made in stroke was that normally there are very low levels of glutamate, just enough to keep our brain cells ticking over. But when a stroke is induced, the levels of this chemical go up really considerably within the brain. They're tens of fold higher than that you would find normally. So whatever's happening, we reduce blood flow to the brain, we reduce our fuel supply, it's switching on a mechanism that causes a massive release of this chemical glutamate in the brain. And you'll note that this occurs really quickly after that stroke has occurred. So now we've identified one harmful molecule, and we know, as I said, that this particular messenger is incredibly important in helping our brain cells communicate to one another, and this all requires some form of energy. So here we have normal brain cell, another cartoon, sorry about this, but it's another cartoon. In fact, my daughter said my illustrations were terrible, so I'll apologise for this in advance. But anyway, this is a cartoon of a normal brain cell. It communicates to other brain cells through this glutamate as a chemical messenger. So when we have a stroke, fuel supplies go down, it keeps control systems that are normally in place, they all break down, and there's an increase or a flood of glutamate in and around the brain cells, and this is incredibly harmful to the brain cells. It also causes a release of other harmful molecules. And so instead of those cells being able to talk to one another, the cell communication goes down, there's brain damage and an impairment of function. So that's really what underlies that weakness that's associated with stroke that you see in patients. So glutamate is bad news, basically. You don't want to have a lot of it floating about, but we need a little bit to keep cells communicating. And so when this was made, this discovery was made, and now this was years ago now, this was at a time when I was actually doing my PhD, it was over 20 years ago. But the reason for showing this is that this was really to lead to a flood of research studies looking at glutamate blockers to see whether by blocking glutamate in the brain they could reduce the effects of a stroke. And as I said, at this time, I was actually working in a lab in Glasgow, and in that lab, they were one of the first labs to develop a rat stroke model. They were working very closely with drug companies. When glutamate was identified, they started looking at glutamate blockers, and were one of the first to show that by blocking glutamate, they could very effectively reduce brain damage after a stroke. Now, these are in experimental models. The glutamate blockers have been given, as I said, quickly after that stroke has occurred. But this and many other studies like this quite convincingly showed that by blocking glutamate, you could reduce the extent of damage in the brain after a stroke. But of course, this couldn't have been good news, because otherwise we'd have had glutamate blockers out there in the clinic. And unfortunately, when these types of drugs were taken into stroke patients, they weren't found to have the same beneficial effects in stroke patients that they had in animal models. Malcolm touched on that in his talk. Now, there are tons of reasons for this failure, but one that I'd like to highlight is that this work was 20 years ago. And we really had very little understanding at that time of the complexity in the way that the brain cells communicate with one another. And if you remember that I said that glutamate is important in cell communication, by blocking all of glutamate's effects, that might have not been the smartest of moves in terms of trying to protect the brain against stroke. Now, this is where we are now. Since that time until now, it's been like putting a jigsaw puzzle together, piecing together all the molecules that are involved in cell-to-cell communication. So this again is another cartoon showing one brain cell connection to another to really illustrate the complexity, all the different molecules that might be involved. It's not up there to bamboozle you in any way, it's to show that the brain is pretty complicated. And actually we need to understand more about how it works in order to really define better treatments. And I'll show an illustration of that now. So now we know that the way that glutamate works can exert both beneficial and harmful effects. And some of these molecules are both harmful and beneficial in mediating glutamate's effects. And so what researchers have been doing now in the knowledge that this is quite a complex signalling pathway, is really trying to identify whether there might be molecules that might be better to target than this broad brush glutamate approach that was done 20 years ago. And so one of the molecules that's been targeted is this protein here. It's called PSD95. Doesn't mean anything except to say that that's been a focus of research in the last few years. And so my group, along with many others, have been studying this particular molecule, and we've used different approaches to see whether by blocking this molecule, whether we could protect against the effects of stroke. And to cut a long story short, yes, it looks that this is quite effective. So we've tried different approaches. We worked with a group here in Edinburgh who've made genetically mutated mice in which this molecule has been removed, and looked at these mice after a stroke and looked at the outcome in mice that had a stroke but contained this molecule, and found that the brain damage after a stroke is markedly reduced in those mice that don't have this particular harmful molecule. From this time, so this is about 10 years ago now, in that time other drugs have been developed which target this particular PSD95 in the brain. It blocks the effects. And if you give this particular drug, it again markedly reduces the extent of brain damage. So these are stained slices to highlight this is an area of brain damage after a stroke here in an animal that's been given a placebo. It's quite a large area of brain damage. If you look at the animals that have been given the PSD95 blocker, then the brain damage is much less. So this looks great, but with a warning sign. This needs to be replicated and reproduced in lots of labs before it goes anywhere near stroke patients. And I think everyone has learned from the mistakes that some of these glutamate blockers that were tested in animal models moved very quickly into clinical trials. Now we know to take things in a much slower, stepwise fashion, to go from smaller animals and move in a stepwise fashion, and a number of recommendations have been put in place such that these types of drugs should be tested in animals that are much more closely related to human. So although the rodents are great models, they're still small, there are differences in the structure of the brain, and it's much better to try these types of drugs in models that are much closely related to human. And last year, a study was published by a group in Canada. They'd looked at this blocker in a primate model of stroke, and they looked at various times after the stroke too, up until a few hours, and then studied the primates many days after that stroke had been induced. And it's to highlight that actually in the primate model, again, this is a placebo, so this is MRI showing a large area of tissue damage. This is primates that have been given the blocker, the damage is much less. And if you look at the pathology, so this is at the end stage, the area of tissue damage is much greater in the placebo group compared to that receiving this glutamate blocker. And importantly, in these models, they've also shown this blocker has also been shown to improve the function, and so the motor impairments that are often seen related to stroke have been improved with this blocker. So this is an illustration how, by understanding better about normal cell function, that this has led to a much more selective targeting of harmful molecules, and studies have gone slowly from preclinical investigation in rodents through to primates, and now there are clinical trials ongoing in this particular area now too. And so this will take a number of years until we know whether it has beneficial effects or not. And so it's an illustration of how by targeting these harmful molecules that are generated after stroke, we might be able to prevent stroke damage. So we can protect the brain against the effects of stroke. But another new emerging area is actually we have our own inherent defence mechanisms in our brain. We have natural defence mechanisms. And work going on in the last few years has really looked to see whether we can take advantage of this defence mechanism. So normally we have very low levels of these toxic molecules produced, but these defence mechanisms that we have can counteract and balance out the effects of these so that we don't see any damage to the brain. However, with stroke, to reiterate, this really causes a huge increase in these toxic harmful molecules. And unfortunately, the defence mechanisms that we have in the brain can't combat these. And so what happens is that we have cell damage. Now, molecules have now been identified that can actually boost our own natural defence mechanisms. And one of these molecules is called NRF2. And there's now good evidence that if we activate NRF2, it basically switches on our own natural defence mechanisms, and it can overcome these toxic, harmful molecules and acts as an inherent protective mechanism. Now, this type of information was actually come from studying vegetables, believe it or not. So many of the food sources that we would normally eat contain substances that are actually NRF2 activators. So NRF2 activators were originally discovered in broccoli sprouts. They're now in Brussels sprouts and carrots and a constituent of curries. But before you all run off and eat a ton of these, which is probably not good for you anyway, you'll never ever be able to activate these defence mechanisms enough to counteract these harmful molecules. You would need to eat tons of them and continually eat them. And so, of course, in the knowledge that we can switch on our own defence mechanisms, drugs and small molecules have been produced which can actually boost NRF2 and stimulate these defence mechanisms in the brain. And in the past few years, there's now a few studies being published which show that these NRF2 stimulators might show some beneficial effects in the animal models. Now, these types of molecules have already been known to be quite effective in other disease processes. So I wanted to highlight this because not only might these be effective in stroke, but there's now evidence to show that they might be effective in multiple sclerosis and motor neuron disease. And one drug company called Biogen IDEC, it had produced a drug called dimethyl fumarate and been studying this drug in animal models of multiple sclerosis. Showed very good beneficial effects of this in treating the signs of MS. And it's now gone from animal models to preclinical trials into clinical trials. And this year it was approved for treating multiple sclerosis. And it's thought that this particular drug acts by boosting these natural defence mechanisms in the body as well as in the brain. And so this year Biogen IDEC signed a 3-year deal with many groups at the University of Edinburgh to study this particular compound, not only in multiple sclerosis but in motor neuron disease and with groups looking at stroke too. So this looks to be a slightly different approach in that this might be a way of treating individuals who might be at risk of developing stroke as well as treating after the stroke occurs too. So this is certainly one new emerging area of research that looks to be promising. So for the latter part, very briefly, what I've focused on up to date is how we can look at what happens to the brain after the stroke and how we might be able to protect the brain against the effects of stroke. But what can we do once that stroke damage has occurred? I've said that it's actually quite challenging to protect the brain. We need to intervene pretty quickly after that stroke has occurred. And so is there any way we can try and promote healing of the brain to try and promote repair and recovery? And unfortunately, in about half of those individuals that will survive after a stroke, they're left with some functional impairment. And so Malcolm indicated earlier that in some of those individuals, they recovered pretty well. So that will be half of them, but the other half are left with some form of impairment. And because the adult brain has a very limited capacity to repair, then researchers have looked at ways that we might be able to, again, boost the repair and the recovery of the brain after an injury has occurred. And so many of you, I'm sure, have heard about the huge potential that stem cells may offer in treating lots of different diseases and neurodegenerative diseases. And this looks to be very promising in promoting recovery of the brain after a stroke. And so the reason that they look to be very useful is that the cells can be given weeks after that stroke injury has happened. The cells can also make the many different cell types that make up the brain. So I've talked about brain cells or neurons, but there are other cell types in the brain, and these stem cells can produce all of these cell types in the brain. And they're also very smart, these cells. They can move to exactly where you want them in the brain. They become integrated into the brain, and they can become functional. And we worked with a company called Neuroregeneration several years ago to look at stem cells they had produced. And in those initial studies, we were really characterising the cells to watch the movement of the cells in the brains and to look at the integration of the cells in our models of stroke. And this is really to highlight how smart these cells are. So we transplanted the cells here, and the area of damage is down here. And I hope you can see that these are fluorescently red, that they're starting to move from this area here down to the area of damage here. So there's some homing mechanism that's telling these cells to move to exactly where they're needed. And moving on from this work, Neuroregeneration then looked to see whether these stem cells could make the types of cells that are required by the brain. And indeed, they showed in a rat model of stroke that they could produce new neurons. They also become integrated into the brain. And in a rat model of stroke, they showed that this impairment that's normally seen after a stroke was markedly improved in the animals that were given the stem cells. And Malcolm has already said that these stem cells are now in clinical trials in stroke patients in Glasgow. And so again, it's an illustration of how, this work has taken many, many years to go from animal models of stroke all the way through to the clinic. And they're still undergoing early clinical trials, but again are showing some promise in promoting recovery of the brain. And of course, we need to know much more about how these stem cells work within the brain after injury too. So just finally, I wanted to close with another new area of research, and it's an area of research that my group is particularly interested in, and that's looking at the links between stroke and dementia. And stroke is will be on the rise, because I'm sure many of you are aware that we're living in a society with an increasing ageing population, and associated with that is an increase in number of individuals with dementia, and actually it's predicted to double over the next 20 years. And this group of remarkable women, or nuns, actually led to a lot of the evidence showing links between stroke and dementia, and they agreed to donate their brains upon their death to medical research. And study of these nuns' brains has really led to new information which has shown that there's tremendous overlap between stroke and dementia, such as Alzheimer's disease and vascular disease. Now, why that happens, we're not quite sure, but in the next few years, we've now been funded to try and understand why a stroke might precipitate dementia, and if by identifying those causes, we then can look to prevent the memory decline in the stroke survivors who then go on to develop a dementia. And so, just to get back to the point that Malcolm made earlier to finish off, my group has worked in preclinical research now for a number of years using animal models. But we've always worked, I have to say, very closely with our clinical colleagues, from clinicians who study stroke patients to neuropathologists, so we can identify changes in the stroke brain. And I think the only way forward really is that we work together and have a collaborative approach to stroke research, because it's the only way really to develop new therapies to try and treat against the effects of stroke. And one other point I would make is that all the work that I've talked about here, I've had many people work with me in the group, too many to mention, but we've been highly dependent on receiving grant or research funding, and that's really what keeps our research studies going in continuity of those. And those are just some of the funders there, and you'll see We actually get lots of money just now from Alzheimer's charities because of this link between stroke and dementia. So I'd like to finish up there and thank you all for your attention and for coming along this evening. It's very nice to see you out. And I'm sure Malcolm and I will take questions. Yeah, thank you.